Namaste. Hello my dear students. Welcome back to online classes brought to you by Vivekananda PU College. I am Supreeth KC. In today's class, we will be continuing the chapter called as cell cycle and division. Okay, whatever the in previous classes we have discussed, from there we will be starting in. Welcome to the online classes again. The chapter is called as cell cycle and cell division. Cell cycle and cell division. Okay. I hope you people are already know the concept of mitosis. What is a cell cycle? Why is a cell cycle? What is the significance of cell cycle? What are the types of cell cycle? Everything you are aware. Dear students, this cell cycle is so important for the organism to survive. Okay? Even for the cell repair, cell replacement, reproduction, for everything the cell cycle is very important. You may be discussing the cell cycle in the form of mitosis sometime it is meiosis, whatever it is, but at the end of the day, everything follows the rule of cell cycle. So, by taking account of mitosis, the knowledge of mitosis, we will be talking about the meiosis too. Dear students, the meiosis, the process of cell division, meiosis, which is very simple and you can compare it with the mitosis, but there are some process that is different from the mitosis. There are some phenomena you can witness which is different from the mitosis. That is the case. So today we are going to talk about meiosis. Okay, yesterday we have discussed detail of mitosis. Today we are going to discuss about meiosis. What is meiosis? Dear students, you already know meiosis is again a type of cell cycle, but how to define this? Meiosis commonly occur, okay, in the process called as gametogenesis. Gametogenesis. What exactly you mean by gametogenesis? The formation of gametes, that's all. The formation of gametes called as gametogenesis. In the process of meiosis, the parent cell, the germ cell, is deployed. Okay? At the end of meiosis, you will be forming cells, okay, which is having haploidicity or n number of cells. If the parent cell is 1, then the daughter cell will be 4, okay, because there are 2 steps here. We will be going to talk about that in very detailed manner. And this meiosis can be termed as, okay, it is a process in which, diploid, it is a process in which diploid germ cell reduce the chromosome number by half in their daughter cell, daughter cells. It is a process in which diploid germ cell is reduced the chromosome number by half in the daughter cell is termed as meiosis. What do you mean by this sentence? Say I have a diploid germ cell, means what? I have a 2n number of a chromosome, okay, In here I have a 2n number of a chromosome, but in the case when the 2n number of a chromosome is undergone a process of meiosis, then at the end of the day, at the end of the cell cycle, I will be receiving daughter cell with the n number of a chromosome. Nothing but the chromosome number is reduced by half. If it is 2n, it is reduced by half, called as n. That's all. The whole process of your meiosis is categorized or divided into two types, two steps. One is called as meiosis 1. Okay. Meiosis 1. The second one is called as meiosis 2. Very simple it is. Meiosis 1 as well as meiosis 2, they are the two steps of the process called meiosis. What is mean by meiosis 1? What is mean by meiosis 2? Okay. Dear students, you already know, in meiosis 1 or in meiosis 2, again there are many steps. Let us discuss that. What are the steps we can study here? Okay, meiosis 1 as well as meiosis 2. Let us see that. Okay. In the case of meiosis 1, see, meiosis 1, there are 4 steps mainly. One is the prophase 1. Prophase 1. Second one is metaphase 1. Metaphase 1. Next one is anaphase 1. Anaphase 1. The fourth one is telophase 1. Telophase 1. Okay, this is of meiosis 1. And again I have, in the case of prophase 1, again I have many steps based on the appearance of chromosome. In the prophase 1, again I can find out 5 categories. Okay, I can find out 5 categories. What are my 5 categories? The first stage which comes under prophase 1 is called leptoten. Leptoten. Okay, the first stage, leptoten. The second stage is called as zygoten. Zygoten. Leptoten, zygoten. Third stage is pachyten. Pachyten. The fourth stage is called diplotin. Diplotin. The fifth stage is called what? It is diakinesis. Diakinesis. So simple. I repeat, dear students. If I take meiosis, I can find out 2 steps there. One is meiosis 1, another one is meiosis 2. In the case of meiosis 1, I can find out 4 steps here. Prophase 1, metaphase 1, anaphase 1, telophase 1. In the case of prophase 1, again there are 5 steps: leptoten, zygoten, pachyten, diplotin, diakinesis. Leptoten, zygoten, pachyten, diplotin, diakinesis. Okay, these are the steps of this process. Then we have meiosis 2, B. Meiosis 2. In meiosis 2, again you can witness 4 more steps which is resembling mitosis there. Okay, A, meiosis. In meiosis, the first stage is called same as of this, prophase, prophase 2. B, metaphase, metaphase 2. Next is anaphase, anaphase 2. The fourth one is telophase, telophase 2. Yes, these are the steps you can find out in meiosis. Okay. What steps are very important, or what are the process which makes this meiosis is different from mitosis? Okay, that is the more technical thing here. In the case of meiosis, dear students, meiosis, okay, here you can find out something called as a recombination, recombination. You can find out something called as a pairing, recombination, pairing, and something called as chiasma, chiasma. These process which makes the meiosis which is very different from mitosis. Recombination, pairing, chiasma. Okay. What exactly mean by this recombination? What you mean by pairing? What you mean by chiasma? We are going to study now. Okay. For the better understanding as well as to conserve the time, I have a chart to discuss. Okay. Where is the chart? Yes. So this is a chart. Okay, this is a chart which is helping us to know the things very better. So here I have written the 4 steps of meiosis 1 are prophase 1, metaphase 1, anaphase 1, telophase 1, right? That you know. And in prophase 1 you have something called as leptoten, correct? Leptoten, first stage. Next you have zygoten, second stage. Pachytene, pachytene. Then deploting, deploting, deploting. Then you have diakinesis, diakinesis. Okay. Let us study one by one in very depth. Okay. What is leptoten? As you know, dear students, when you take a cell cycle, first you have a G1 phase, then you have a S phase, then you have a G2 phase, then you are getting into M phase, right? This is already you know. G1 after S after G2 after M. In G1 phase, the cell or the cytoplasmic component of the cell is increasing, as well as you can witness a little heavy mass of a cell, the size of the cell becoming larger. In the case of S, where nothing but it is known to be the synthesis phase, you can witness the DNA synthesis or the recombination, replication of DNA you can find out there. And in G2 phase, of course, you can find out the presence of protein, synthesis of protein, which is very important for the cell cycle or the cell division. In a M phase, nothing but your meiosis is happening. But you already know in the S phase, the whole content of a DNA is doubled. Now the chromosome number, the content of the DNA is doubled. Okay. Next we are entering to M phase, nothing but the leptoten phase. In the case of leptoten, okay, in the case of Leptoten. Sorry, that is by mistake. Leptoten here, you can find out, no? See, I have a leptoten phase here, zygoten, pachytene, diplotin, then diakinesis. This is called as a prophase 1. After prophase 1, I have a metaphase 1, anaphase 1, then into telophase 1. Able to see, right? Okay. Fine. This is a common cell. This is a normal cell where it is enveloped by the cell wall. Then you have a nuclear membrane which is enveloping your nucleus. Inside you have a nucleolus. Then you will be having a chromosomal material, or it is called as a chromatin fibers. Correct? Then all the cytoplasmic component you will be having. This is a normal cell. Done. Okay. When a normal cell passing from G1 to S, S into G2, G2 into M, it is actually entering to the M phase, right? Where the meiosis is happening. Now the cell is ready for meiosis 1. In the meiosis 1, it is a prophase 1. In the prophase 1, the first stage is called as leptoten. Yes? Now, see, this is the diagram which defining the first phase of a prophase called as a leptoten, where the chromatin fibers are becoming a little longer and it is having a shape of slender shape, and the nucleus is enlarging. Look at here, this is the nucleus, right, which is enveloped by nuclear membrane. This is getting a little larger, enlarge. Okay. And the chromosomal fibers or chromatin fibers, this one, is in the shape of a slender, as well as it is a little longer, shape becoming little slender as well as longer. So, in the case of leptoten, you can find out long, slender chromatin fibers. Chromatin fibers. Long, slender chromatin fibers. Okay. After this, the whole set is ready to enter the zygoten phase. Okay, zygoten phase. Now you check out, even when the beginning of a prophase taking place, the aster which is present in your cell, they undergone a copy, and you can witness 2 copies of a aster here. Okay. Then it is entering to the phase of a zygoten, where the chromosomal material, whatever you are witnessing in leptoten, they become more condensed form. Condensed chromosomal material you can find out. And the most important thing is, in the case of, or in the phase of zygoten, okay, the similar chromosomes, whatever you can find out inside the nucleus, are getting a pair. They are pairing up. The similar chromosomes are pairing here. The similar chromosomes which are pairing, similar chromosomes paired up. These similar chromosomes are called as a homologous chromosome. Homologous chromosome. What do you mean by homologous chromosome? The chromosomes, pair of chromosomes which are having higher similarity in the size or in the place of centromere, even the gene loci. Okay, if they are finding out a similarity, then they are known to be homologous chromosome. They will be paired up. See, homologous chromosome from the parental side, from the maternal side, Okay, mother side as well as the parent side, father from the father side, they will be paired up. Chromosomes are paired up. Okay, when they pair, the point of a pair is called as synapsis. Synapsis. When they pair up or when they attach each other, called as synapsis. Now the structure is called as synaptonemal complex structure, with the showing synapsis called as synaptonemal complex. Synaptonemal complex. Synaptonemal complex. That is called as synaptonemal complex. Yes? Okay. So we are witnessing the synaptonemal complex in zygote, correct? Synaptonemal complex is over and even homologous chromosomes are paired. Okay, homologous chromosomes are paired. Homologous chromosomes are paired. Okay, they are lining aside by each other. They are lining aside by each other. Homologous chromosomes are paired like this, like this. Okay. After this, we are entering to the phase of packeting. Now after zygote to packeting, when homologous chromosomes are homologous chromosomes are paired, okay, then even condensation is happening there. After that, the whole structure is looking as a tetrad. This one, whole structure looking as tetrad, because you will be having, because of the condensation, you can find out the sister chromatids too. Okay, chromatids with another two arms, sister chromatids. So this structure is known to be the tetrad, because homologous chromosomes are paired up here. See, look at there, look at here, look at here, look at here, and even you can witness sister chromatids also. Yes, sister chromatids also. Now in this stage called as a tetrad, like this, chromosome, these are known to be the homologous chromosome. Now I don't have a color chalk here. Now, okay, imagine this is a colored part, half will be colored part, this is blank. They will be attached to each other side by side, and they are looking like this. This is one chromosome and this is another chromosome. Okay, like this. This stage is called as a tetrad. This stage is called as tetrad. Now, at the point of attachment of these chromosomal arm in between non-sister chromatids, these are sister chromatids, chromosome 1, chromosome 2, these are sister chromatids. Okay, in between non-sister chromatids, here and here, this part and this part will be linked up, this part and this part will be linked up. Okay, now what happens here, these are known to be the non-sister chromatid. At the point of attachment, here attachment will be happening. At the point of attachment, okay, the exchange of genetic material will take place. From here to here, here to here, the exchange of genetic material takes place, resulting recombination. Okay, look at here. The same structure what I am discussing here as a tetrad, this is a structure of tetrad. in between the non-sister chromatids, see this is different, this is different, in between those two there will be a point of contact, there will be a point of attachment where the exchange of a genetic material will take place. Look at the structure. Here one chromosome is they defined as in white color. Don't look at this. Okay, this is happening in packaging stage. See, this is appearing one color, this is appearing another color. Both chromosomes are appearing different color. And at the point of a contact in between the non-sister chromatids, where genetic material exchange will take place, leading to recombination. Okay, recombination. Look at here. Okay, in this place where the recombination is happening, okay, that is actually by, okay, crossing over, that is known to be the crossing over, where the recombination is happening, where the genetic material is exchanged in between, okay, there recombination is happening. That process is called as a crossing over. See, this is one chromatid of one chromosome, another chromatids of another chromosome. Both will paired up, and here whatever the genetic material is acquired by one chromosome will be exchanged to this arm, and whatever the genetic material acquired by this chromosome will be shifted to this arm. Okay, this is how genetic material exchange will take place, known to be the crossing over, leading to the recombination, which is happening in packaging stage. Okay, after this the whole setup, okay, whole setup is called as a chiasma, a region of chiasma where recombination is happening. Phenomena is called as a crossing over. Okay, then we are shifting into the deploting stage, where in deploting stage, here I write, dissolution of synaptinymal complex takes place. Synaptinymal complex takes place. But before going to deploting stage, I must tell you where the recombination is happening or the crossing over phenomena is taking place. The exchange of genetic material is led by the enzyme, or the whole process is triggered by the enzyme called as a recombinase. Okay, recombinase enzyme. Done. In the case of next stage of deploting, okay, deploting, where the dissolution of a synaptinymal complex takes place, nothing but, see in this structure you can find out, okay, here you can find out, witness the closed structure of a centromere, and this centromere will be pulled apart, okay, and only at the point of contact of recombination, where the recombination is happening, okay, only point of contact here only, but the centromere part will be pulled apart, okay, and it is looking as X-shaped structure. Only chiasma region, point of contact, will be held together tightly, and this one is moving apart. See, here you can find out this structure called as chiasma is only leaving behind, and the centromere will be pulling apart. So that we are at the stage of deploting now, pulling apart, leaving the chiasma, and the structure is looking as X. Done. Okay, next we are entering to the diakinesis, where the terminization will be taking place. Terminization is nothing but, even where you can find out the whole chiasma, whatever we are referring here, whole chiasma, only the point where we are left behind in the stage of diakinesis will be even pulled apart, and the structure they become separate, whole structure becoming separate, whole structure will be becoming separate. This is the ending story of your prophase 1, nothing but the diakinesis, last stage of prophase 1. Okay, here the nucleolus is completely disappeared. Okay, then even you can find out the position of your centrioles, asters will be moving apart to the poles. Okay, they will be moving apart to the poles and ready for the next step. They will be moving apart. Now the whole structure is stabilized. This asters, okay, is stabilized. You can find out, no? The whole structure is stabilized by the formation of some fibers or microtubules. Whole structure is stabilized by the microtubules known to be the spindle fibers. Spindle fibers. So the centrioles will be coming to the poles, opposite poles, and also the whole structure is stabilized by the presence of microtubules called as spindle fibers. Now we will be entering to the next phase of a prophase called as metaphase. Metaphase 1. Of course, in metaphase what will happen? The whole set of a chromosome will be aligned to the equatorial region. Aligned to equatorial region, nothing but if the cell is like this, you can find out the chromosome here and it is appearing like this. Okay. So the structure of formation of a metaphase plate will be stabilized by the support of spindle fibers. Even you can find out the chromosome. Okay, and here the centromere, which is holding your chromosome, which are undivided. Okay, you have a chromosomal arm as well as a centromere which is undivided. Okay, next stage, but in the stage of anaphase, what will happen means centromere will divide. Okay, centromere will be dividing to each other. Okay, which is a very important stage here, where when the centromere is dividing from the homologous chromosome, okay, which is undergone a recombination, and even you have a presence of non-sister chromatids content. Okay, when the centromere is divided, the recombinant chromosome will be pulled apart. Recombinant chromosome will be pulled apart to the both side by the spindle fibers, and they will be reaching their respective poles. They will be reaching to their respective poles. Okay, when they are reaching the respective poles, now the cell division is done. Now what should happen? Okay, there will be a wall formation. In the case of animal furrow or cleavage furrow will be happening in the case of animal, cell plate will be happening in the case of cell. From the wall, from outside to inner side, the wall is protruding in so that it can divide the cell into different structure. Where the chromosomes are decondensed, as well as envelope will be forming, nuclear envelope will be forming, and cell will be entering to the normal state. This is the state of first telophase, and next it is followed by interphase. Here the cytoplasmic material also divided, so cytokinesis is over. Then we are entering to the interphase. Interphase is a state where the chromosome or the state in between the successive meiotic division, or meiosis 1 and meiosis 2, the stage in between meiosis 1 as well as meiosis 2 is called as interphase. Okay, so the meiosis 1 is done. Meiosis 1 is done. Once please carefully observe this chart, okay, for about a minute. Please observe. This is very important. When the life of meiosis 1, when it starts, the chromosome number will be 46 here. Okay, 46. Even the content is doubled there. You have another copy of 46. Copy. Number of a chromosome is 46 only, but you have a copy. Okay. And this copy of chromosome, what you have here, they will be separate such a way that when the homologous chromosomes are attached, and even when they are segregate, or when they reach to their opposite poles, okay, the structure is like this. You have 46 number of a chromosome. When they are reaching here and here, you have a copy of a chromosome. You have copy of a chromosome. The whole 46 chromosomes, when the homologous chromosomes are attached, and as well as, sorry, not here, as well as when they pulled apart, they will be pulled apart to the opposite pole. Homologous chromosomes are pulled apart to the opposite pole, as well as when they pull over to the opposite pole, you have a copy of chromosomes here. You have a copy of chromosomes. Okay, 23 pair of a chromosome along with a copy of itself will be conserved in a first telophase. 46, okay, 46 copy, they will be dividing to each other, 23, 23. 46 were with the copy, right? Content were doubled. Now again you have a 23, 23 copy with its 23, 23 pair with its copy. So you will be having 23 chromosome with the copy. 23 chromosome as well as 23 chromosome with the copy, and this is called as n number of chromosome. N number of chromosome. This is about meiosis or first meiosis. So thank you so much. By the way, this much is enough for you today. Thank you so much for your patient listening, and we have discussed meiosis, first meiosis today. I hope you people are cleanly understood the concept, and you people are staying at home. Stay safe. Okay, stay at home, take care, have a nice day, and read well. Okay, thank you so much for your patient listening. Have a nice day. Thank you.